This lecture covers the really cool topics of chemotaxis and motility. And the two obviously go hand in hand because microbes don't just move around at random, despite what it looks like under the microscope when you're doing like a hanging drop in micro 224. The microbes obviously have a very sophisticated mechanism for sensing their environment and going towards nutrients and things like that and away from things that are toxic. And they also have a mechanism for remembering how much of some concentration of a certain chemical is present and whether or not to go in a certain direction to find more of it or change directions. So this slide is an overview of flagella ultrastructure. And so you can see on the left the typical gram-negative arrangement and on the right the gram-positive arrangement. And so in this particular case, the gram-negative is an outer membrane, and embedded in that outer membrane is an L ring, the L standing for LPS, or lipopolysaccharide, and the peptidoglycan layer here in the middle, the thin peptidoglycan layer with the P ring, or peptidoglycan ring, embedded. And then here in the plasma membrane, the S and M rings embedded, standing for supramembranous, meaning kind of lying on top of the membrane. And then through the center here is a rod, and then this bend called the hook, and then the filament itself, which is the flagellum. Over here with the gram-positive arrangement, you see the thick peptidoglycan layer, and then essentially an S and M ring embedded in the, or sitting mostly on top of the plasma membrane, and some of it embedded in the plasma membrane. This is a look at a little bit more detail, and then over here some actual experimental work. And so we can see the filament and hook and the L and P rings of this gram-negative bacterium. But the added features are these stator proteins, the motor proteins that you see here, mode B and mode A, and they're referred to as stator proteins because they don't move. What they are is basically motors that operate through proton motive force. As you can see, as protons move past these, that energizes this motor and causes the S and M and C ring that you see here to rotate. And in turn, that creates torque and turns the filament to make the microbe move. And embedded in here as well are the proteins for a type 3 secretion system, which the microbe uses to secrete out of this structure. So not just filament proteins either, but other things as well, as you'll see. Over here are some actual data, as I mentioned. And so again, this is Vibrio fischeri that we've been working with a fair amount in the lab. And this is the wild type. So here's the bacterium and then a tuft of polar flagella. A friend of mine made this FLA mutant. So FLA is one of the main filament proteins. And so what was interesting is when she knocked out the FLA gene, we assumed that the bacterium would be completely non-flagellated, but it turns out that they have at least a single polar flagellum, suggesting that other flagella proteins are required to make these flagella. Obviously, FLA is the big one because you can see that you go from a tuft to a single, but you don't eliminate the entire flagellum, again, which is interesting. And then this is just a spot plate. We'll do this in lab where you can spot different strains of bacteria and look at their motility phenotypes. So you're spotting these on soft agar, something that has less than the 1.5% agar that's typical for a hard plate. Usually we use 0.25% agar or something like that. And you just spot the microbes on there. And because it's wet and soft, the microbes can actually move across the surface according to their abilities. And so here's the wild type spotted here and then moving and creating these rings of migration. Whereas the FLA mutant, having only the single polar flagellum, can barely move in the same time frame. This is a closer look at a few things, one of which is the C ring and how it interacts with the rest of the basal body here. But also the other thing I wanted to show you is the type 3 secretion system that sits in the middle here and then can grab flagella proteins and so forth and kick those out to add them to the growing filament. The other thing to notice here is just some gene regulation, and I'll talk a bit more about this in a few slides from now. Different types of microbes, their organization of flagella. So Caulobacters have a polar monotricious flagellum like this. Vibrios generally have a polar monotricious, but as I showed you here, Vibrio fischeri has a polar lophotricious, meaning a tuft. But cholera looks more like this. Wild type cholera looks more like that, with a single polar monotricious flagellum. That's why that's on here. I think they're referring specifically to cholera. And then Salmonella has a peritricious type flagellum, like you see here, the flagella emanating from the entire perimeter of the bacteria. This is showing you the promoter of the bacteria. The bacterial genes. And so whether or not it's sigma 73, sigma 54, or sigma 70, these sigma factors assist in gene regulation, as I'll show you, and then the master regulator itself, these transcription factors that are essential. Again, and I'll show you some of that in a few slides. So a lot of that is summarized here, the coupling of flagella gene regulation to flagellum assembly. And so we start off here looking at the different classes of flagella genes. So class 1, these particular genes are the FLHDC operon activated genes. And so they again, these are a variety of transcriptional activators that work with this sigma factor called sigma 70. So these transcriptional activators work with sigma 70, which associates with RNA polymerase, to drag these transcriptional activators and RNA polymerase to the correct promoters for these class 2 operon genes. These are the so-called HBB genes for hook and basal body. And so you can kind of see here in the cartoon what's being made at each step. There's a whole bunch of genes, 9 operons comprising 38 genes, and then these 2 regulatory proteins fall in the class 2. And so all these structures here are synthesized by these 38 genes, the hook and this basal body motor. One of the things that's made in this particular class 2 set is fly A, which is called sigma 28, as you see here. They're color coded as well, and flag M, which is an anti-sigma factor. And so, as you can see, flag M will bind to sigma 28 and keep it silent. And then, when the time comes, sigma 28 can be released flag M will release from sigma 28 and actually be secreted by that type 3 secretion system. And when that happens, sigma 28 is free to activate, to join forces with RNA polymerase and activate the class 3 genes. And so the class 3 genes are 16 operons comprising 30 genes. They make things like fly C, which is a major filament protein. They also add these motor force generator proteins, and they also add hook filament junction proteins, and then add to the filament itself in addition to producing cap proteins. And so again, this little step here is really important as the organism proceeds in the production of hook and basal body motor. When this structure is complete, there's got to be a signal to facilitate the completion. And as a result, that signal is typically the flag M dissociating with sigma 28 and then getting secreted from the cell so that 28 can, again, bind to RNA polymerase and do its job. This slide overviews repression of Vibrio cholerae HAP-R during mucosal penetration. So essentially what it's doing is showing you the relationship between flagella rotation and virulence. And so in this particular case, cholera might be ingested into the small intestine, but in order to get there, it's got to go through the acidic stomach and then the shower of bile in the duodenum. So a lot of the cells are destroyed. And as a result, the bacteria come in pretty low cell numbers. That low cell density basically results in an activation of LUXO which will block HAP-R. HAP-R is I should point this out, is a homologue of LIDAR in Vibrio fischeri. So this is Vibrio cholerae's version of LIDAR. But in this particular case, it controls virulence, whereas in fischeri, LIDAR primarily controls light. So in this particular case, so bacteria penetrate mucus. So once they get into the small intestine, they've got to get through the mucosa, and they've got to swim to the small intestinal epithelial layer. And so while that's happening, the flagella are rotating and breaking. And when their flagella break, when the flagella break off, that results in a structure like this, basically putting this microbe back into this state where the type 3 secretion apparatus starts grabbing on to flag M and secreting it, freeing up sigma 28 to bind to RNA polymerase and activate the class 3 late genes to make more filaments. And so one of the other things that fly A or sigma 28 does, in addition to activating flagellins, is it blocks HAP-R. So by taking HAP-R out of the mix here, you basically, so again, you're removing a repressor of virulence factors. Once you remove that repressor, cholera toxin and TCP, the toxin co-regulated pilus, are activated essentially. So this cascade of transcription factors is freed up to activate the toxin and this pilin protein that allows it to adhere to tissue and to the cells. And so again, the cell becomes more motile and more virulent. So chemotaxis is movement towards a chemical attractant or away from a chemical repellent. And the concentration of chemical attractants and repellents is detected by chemoreceptors on the surface of the cell, so basically embedded in the cell membrane. And that's pictured here. So what we call them are methyl-accepting chemotaxis proteins, or MCPs. And this is E. coli. This is just a few of them in E. coli. You can imagine that it's got a whole network of these MCPs that recognize all kinds of chemicals in the environment. So here's one called TSR that's specific for the amino acid serine. And here's one called TAR that's specific for aspartate, the amino acid aspartate, and potentially the disaccharide maltose. And here's one called TAP that non-specifically recognizes dipeptides. So, groups of 2 amino acids, peptide bonded to one another, or TRG, which recognizes galactose, the 6-carbon sugar, or ribose, the 5-carbon sugar. There's even aerotaxis proteins, AER, that recognizes oxygen levels. And just below that are these proteins called chemoattractant proteins called KW and Ka. And these are basically what transfer the signal to the rest of the chemosensory system, as I'll show you here. So here are the extracellular molecules. They can be sugars, amino acids, whatever. And here are the MCPs, the chemoreceptors. And so these chemoreceptors, once they've interacted with some kind of ligand, will signal to the sensor kinase here, KW and Ka, to transfer phosphate groups or not transfer phosphate groups readily to KY, the response regulator. So this is what's called a 2-component system, as you see here. The first component is the sensor kinase, KWA, that actually senses the signal, well, indirectly through the MCP, and the response regulator, which actually interacts with the target, in this particular case, the basal body motor. These guys here, KR and KB, this is a methyltransferase, and this is a methylesterase, so they sort of compete with one another to methylate the MCP, and I'll show you that. Put this all together in a minute, but I just wanted you to see the overall structure and the main players. So here's another picture of that, but before I get into it, I'll do this. I'll take my own advice and show you the animation. So here's a bacterium in the absence of a concentration gradient, just kind of tumbling and running. Every one of those little dots is a tumble point. You see the microbe tumbling where it's going, where it's not moving in any particular direction. It just sort of spins in place and then runs for a short period of time and then spins again and runs. But the general gist of it is that the microbe doesn't really go anywhere. It just kind of goes in a loop. And that's mostly what you see when you're looking at microbes under the microscope. Again, for example, in micro 224, when you do a hanging drop, the microbes just kind of look like they're kind of going in no particular direction, and they aren't really. There's no concentration gradient. They're in a rich medium, and food's everywhere, and so they're not really worried about rushing to the highest concentration of this or that. But in this next cartoon image, you see the microbe tumbling and running, but the frequency of runs to tumbles has increased so that the microbe is actually going somewhere in a particular direction. It's not perfect, it's not an exact straight line, but the microbe has gone from here to here. And in the process of doing that, has moved a little bit, tumbled a little bit, and what it's doing is processing the information in the environment. Is this environment more favorable than this environment? And the answer is no. If the source of your positive chemical is here, the answer is no. So the microbe spins again and tests out this in this direction, and then decides, is this environment better than this environment? And if the answer is yes, the microbe will run again in this general direction, and so forth until it gets to where it's going. So hopefully you can see that this is basically a form of memory, and this is sort of a primitive brain, if you will. So this next sequence is illustrating the MCP, methyl accepting chemotaxis protein complex, as well as the sensor kinase complex that they'll show you in a minute here. There's key W and key A. So there's the sensor kinase complex interacting with the cytoplasmic side of the MCP. So as you can see here, when the MCP is not bound to an attractant, it stimulates key A to phosphorylate itself, or autophosphorylate, using ATP. Key A autophosphorylation is inhibited when the attractant is bound to its MCP. So in this particular case, there's no chemoattractant. The MCP is not bound. Therefore, key A readily autophosphorylates. So grabs the phosphate off of ATP and shuttles it to the response regulator that you'll see in a second. So there's ATP, autophosphorylation. Key Y, the response regulator, grabs that phosphate and interacts with the basal body motor. Key Y phosphate interacting with the basal body motor stimulates clockwise rotation. Clockwise rotation stimulates tumbling. So not surprising, if there's nothing bound here, then this event happens very frequently, and therefore you get frequent tumbles, and therefore the microbe doesn't really go anywhere. But key Y phosphate doesn't just stick to the rotor like that. It basically has to be removed periodically. So it rapidly binds, and then it's rapidly cut by key Z. And so key Z will cut that, and momentarily the flagellum will go back to counterclockwise, and the microbe will run. But because there's nothing bound here, this is happening quicker. There's lots of key Y phosphates ready to bind, and key Z can only work so fast. So the signal to rotate the flagellum clockwise is rapid, and therefore again the microbe will mostly tumble and hardly move anywhere. So now what happens when a chemoattractant binds? Chemoattractant binds. Key Z is not very good at autophosphorylating under this circumstance. And as a result, there's not as much key Y phosphate coming over to tell the flagellum to rotate clockwise. There's a much greater frequency of counterclockwise rotation. So the microbe actually starts going straight for longer runs. The other question is, hopefully you have, and I alluded to about that brain mechanism. How does the microbe know that one environment has more stuff in it, more good stuff in it than the previous environment? And so, and because again, the microbe doesn't want to just keep going straight and run past everything. So the microbe actually has to remember essentially. And that memory, that primitive memory, is based on methylation, as you see here. This key R, the methyltransferase, is constantly adding methyl groups. And another protein called key B that I introduced you to before, the methyl esterase, is constantly removing them. And so under this circumstance, key R is doing its job and methylating. And if this guy can autophosphorylate quickly, meaning that there's nothing bound here, if this guy can autophosphorylate quickly, as it does in the absence of a signal, then key B will just pluck off the methyl groups as quickly as they're added, and therefore the microbe will be in a state of, essentially a state of stasis. So there's the MCP being bound. So when the attractant is bound to MCP, the complex becomes a good substrate for key R. So that means that the microbe wants to run more now, right? And so key R starts winning. Key R starts methylating the heck out of the MCP. Key A can't keep up because it's not able to autophosphorylate as well. So key B is not as active and therefore is not taking the methyl groups off. So really what the cell is doing is measuring levels of methylation of the MCPs. If they're highly methylated, then that would favor runs. And don't forget that again that key Y is is part of the reason why this is happening. It's not just this overmethylation, but key Y phosphate is in lower concentrations as well. So the default setting here is for counterclockwise run. And of course the opposite then happens, where key B will win if there's no chemical attractant bound. And of course, more frequent key Y binding will continue the clockwise rotation. And so this is just showing you that again, the key W, key B, key Z, all these guys, and giving you a little bit more fancy look at the motor proteins. And again, here's another look at it. So I'm not going to get into that. Now that you've seen the video, you can kind of use this if you want. This is just an electron micrograph showing what an individual had done using specific antibodies to these proteins here bound to gold particles, just to show you where they all concentrate, typically at the poles of this rod-shaped microbe. And again, this is just another look at that with a little bit more detail. So again, you can see the flagellin proteins, Fla A, Fla B, Fly D, the cap, the hook protein, flag E, the rod, and then the L and P ring in this gram-negative. And, again, the export apparatus, the type 3 system, the stator proteins, mote A and mote B, the, proton motive force, the ring C, and so forth. And here's the MCP and key W, key A, key Z, key Y, key R, and key B. So again, just to kind of just different looks at this. Again, it's not new information, shouldn't be new information, but it's just a different look at this for you to help with your studying. And I already went over this, the concept of run and tumble, that was covered nicely in the animation. Here's other types of motility. So that was all obviously flagella-based motility, but in this particular case, we're looking at spirochete motility using axial filaments. So spirochetes are like, syphilis and bacterium that causes Lyme disease, Borrelia. And so these are organisms that have axial filaments, or so-called periplasmic flagella, running through their periplasm. They have basal body motors and everything, but they're bound internally to the bacteria. That's not what I wanted. There we are. So in this particular case, you can see that the here's the where the basal body motor would be, and that would cause a rotation, that would cause the barrel of this microbe to rotate like a corkscrew. And so again, these are syphilis bacteria and Lyme disease, the spirochetes. And so this type of corkscrew motility really gives the microbe the ability to penetrate really deeply into tissues. One of the reasons why Lyme disease is really difficult to get rid of. The microbe can penetrate into joints and so forth and cause really long-lasting arthritis and autoimmune problems. Another thing to look at here is the so-called twitching motility that you see, that I've mentioned here. And this is just the ability of microbes to move along surfaces using very specialized pili. And so in this particular case, you can see Pseudomonas aeruginosa twitching, moving along a surface. And this surface could be an agar plate, or it can be the lungs of an infected patient. And so again, they're not using flagella here. They're moving as a result of this particular type of pilus. Another version of this is illustrated here, where the microbes are actually, in this particular case, walking. And so here are these bacteria, and they have these pili on one pole, and that the microbe stands up essentially, and by retracting the pilus is able to essentially walk, which is pretty fantastic. Oops. And so again, this is the periplasmic flagella, just a nice diagram of that with the basal body motor winding throughout the periplasm, and then again attaching to the back end. So as these motors rotate, that places torque on the barrel here and causes this microbe to corkscrew. And so again, here's that what I mentioned already, the bacterium, and then it puts out these pili that they're able to hook onto something, and then by retracting this pilus allows the microbe to drag itself. And that's what was happening in that video previously, and the walking video too. The ability of these to basically retract in order to make this guy look like he's walking. And so here's a nice representation of one of that type of a pilus that facilitates this walking behavior, or again twitching behavior. So here's the outer membrane, and then the cytoplasmic membrane, and again you'd have the periplasm and cell wall in here. And so here's the main pore, pill Q, that forms, that allows the pillin proteins to be projected through. There are a bunch of minor pillin proteins that attach to pill BC. So here's pill BC at the base, and then these minor pillin proteins which help pill A to assemble at this base. So these pill A proteins are the main pillin proteins, these purple little spheres here. Pill T is a retraction ratchet, essentially. So that helps pull the pilus back in. And then pill B is essential for assembly, so ATP binding and hydrolysis. So that ATP-driven process allows these guys, using this as a scaffold, and this is a scaffold, and this is a scaffold, to assemble these pill A proteins to quickly put out the pilus, and pill T to quickly retract the pilus. So one of the cool aspects of this, not just movement, as I showed you, because this retraction event is essential for movement. I mean, it's not just the retraction; it's the assembly and retraction that causes the movement. It's like throwing out a grappling hook or something like that and pulling yourself up the side of a mountain. But pilus retraction can also lead to DNA uptake, and so. And again, this concept of transformation, I've already alluded to that in the genetic variation slides, that microbes need to have DNA uptake machinery. Well, this is the DNA uptake machinery in greater detail. Here are the gram-positive bacteria, and they use these COM proteins that allow them to elaborate a COM-GC pilus into the environment. And then imagine this COM-GC pilus, grabbing onto free DNA in the environment, kind of like twirling your fork around some spaghetti. When this thing retracts, it brings the DNA into the cell, and then the microbes can use competence proteins and nucleases to cut the DNA into a single strand and thread it through a pore, COM-EC pore, and COM-FA as well. These are, DNA binding proteins, pores, and ATP binding and hydrolysis proteins to provide energy, to ratchet that DNA into the cell. The gram-negatives are using essentially the same concept, but they have an outer membrane, so they have the pill Q that I already showed you. And then the major pillin proteins, and then the base proteins, and again they're named differently for different organisms, so hopefully you don't get confused by that. Here's pill C, B, and T. In this particular diagram, there's pill G, pill F, so they're the same proteins but just different names and different organisms. But pill Q is the same anyway, it's the pore. And so when this thing goes out into the world, is assembled and then retracts, it can bring that DNA in thanks to these guiding proteins and pores and ATP binding proteins and nucleases. So this process is linked to cell density. So here's a low cell density environment. And in this particular case, the microbe in this low cell density environment is pumping out a nuclease, which allows it to chop up DNA and degrade that DNA, basically to use it for food so it can grow. But when it's in high cell density, the microbe doesn't necessarily need nutrients anymore. Now what it wants to do is start, undergoing evolution. And so in this particular case, it will shut down its nuclease and favor natural transformation. So favor the production of the competence proteins and the pillin proteins to assemble those on the surface, and then grab some of this free DNA, bring it into the cell, and then recombine that DNA into its genome for, like I said, natural evolutionary processes.